All right, hey everyone. So we are now on video 4.2, and this is focusing on specifically the cell membrane of our cells. And the cell membranes are actually quite complex, and there's a lot going on. So we have a video dedicated to the cell membrane. So the first thing that we're going to do is actually we're going to watch this video, and then I'll talk about it. But let's just watch this video. All about bioluminescence.
These waves, glowing mysteriously on an August summer night, are just off the coast of Monterey Bay in California. It's a phenomenon you might even have seen yourself, an ocean lit up at night with countless tiny flashes. But what causes this striking phenomenon? Where does the light come from? Bioluminescence is produced by an incredible range of organisms. Some bioluminescent organisms, like fungi, are found on land, but most of them live in the ocean. For bioluminescence, creatures require two unique chemicals, and here's how it works. The enzyme luciferase causes a chemical reaction. Luciferin molecules react with oxygen to absorb energy and finally release it as light. Although different forms of luciferin can produce different colors of light, marine organisms emit blue light. This might be because blue light can travel the farthest, while water absorbs other colors more quickly. Throughout history, stories of bioluminescence were often thought to be pure fiction, and seafarers have been telling each other magical tales ever since. But in 1995, the captain of a British vessel wrote a real-life account in his ship's log.
At 1800 hours, on a clear moonless night, while 150 miles east of Somalia coast, a whitish glow was observed on the horizon, and after 15 minutes of steaming, the ship was completely surrounded by a sea of milky white color, with a fairly uniform luminescence.
Reports like this are pretty rare, and there was no photographic evidence. Some scientists, including marine biologist Stephen Haddock, were curious and sought confirmation from above.
We wondered if you could find one of these ship reports where they record sailing through one of these milky seas and actually find the corresponding satellite data that cover that area at that same time. So we looked at this satellite from the ship report in 1995, and it was somewhat of a eureka moment. We cleaned up the noisy sensor image from the camera, we mapped it onto the ship track, and this 300-kilometer feature emerged on the map, matching exactly with what the ship had reported. So it was really an amazing moment.
And so satellite images from space did validate the story in the ship's log. But what was causing a glow so bright it could be seen from space? There are many different bioluminescent marine organisms found throughout the ocean. One of them can even be found at the back of an ordinary fridge. Left for a couple of days, this sea bream starts to glow. The fish itself doesn't have any light-producing abilities. The glow is in fact produced by the bacteria found in almost all seawater when they start to feed on decaying fish. When currents and temperatures cause a large bloom of algae in the ocean, these bacteria also feed on dying algae. Once they reach a critical concentration, their secretions trigger light in others too. Eventually, they glow in such numbers that they can be detected by a satellite in orbit. Bacteria might have been the very first living things to glow, but they aren't the only reason why the sea lights up. If you put a drop of ordinary seawater under the microscope, you can see it's full of tiny organisms invisible to the naked eye. These are dinoflagellates. Dinoflagellates are one of the biggest single-celled organisms known to man. They're neither bacterium nor plant nor animal, but they do share some characteristics with both plants and animals. 90% of all dinoflagellates are marine plankton, and when conditions are right in the sea, they bloom in enormous numbers. Left alone, dinoflagellates won't produce much light, but if something like an ocean wave, a current, or a human disturbs them, they will flash brightly. This phenomenon was visible in August in Monterey Bay, where the waves off the coast glowed blue. According to the Monterey Bay Aquarium, a mass bloom of dinoflagellates caused this bright light. The event occurs every few years along the coast of Southern California. But locals say this year's sea sparkle is especially vibrant, possibly related to the historic rains that soaked the region and generated algae bloom. Bioluminescent tides like this are rare, but dinoflagellates are found in huge numbers all over the world. They're among the most widespread of all bioluminescent life. Wherever they exist, these single-celled creatures illuminate anything that moves, like these bow-riding dolphins. The chemical reaction that emits light requires a lot of energy, so there needs to be a benefit to the organism that makes it worth their while. If you're a single-celled organism like a dinoflagellate, you're at the bottom of the food chain, so avoiding getting eaten involves hard work every day. Scientists think that bioluminescence could be a defense mechanism to help them escape predators. Imagine a shrimp comes along and starts feeding on dinoflagellates by filtering them out of the water. The shrimp will be illuminated as it feeds. This could attract the attention of predators, which might in turn feed on the shrimp. Just as a flashing burglar alarm alerts police to a thief, the dinoflagellates expose their attacker to its enemies. In this case, a cuttlefish catches the shrimp, and the dinoflagellates don't need to be afraid anymore. There are many other bioluminescent organisms besides bacteria and algae.
All right. Okay. So that was, we went over that example, or we watched that video because how is it that they're able to do this bioluminescence? Well, it's because there are these enzymes called luciferase that are found and embedded in the cell membrane, not in our cell membrane, but in organisms that can do this bioluminescence. They actually have these enzymes, and there's a chemical reaction that occurs. A luciferin will interact with the luciferase, and it creates this bioluminescence. So another example, they didn't go over an example like this, but was the firefly squid actually also has the ability to have this bioluminescence. And for the firefly squid, they actually will hang out kind of near the surface of the water, and by glowing, it actually, and there's the night sky, by glowing, it actually is a camouflage mechanism for the firefly squid. And then this is just another, I just thought it was a nice image, but they kind of talked about this idea when phytoplankton or other organisms that are agitated, like the dinoflagellates, when they're agitated, they produce this bioluminescence, and many times it is a defense mechanism. It exposes the predator. So just thought that was a neat way to kind of introduce that there are these enzymes that can produce this light emission, and it's found within the cell membrane. So when we're looking at our cell membrane, we want to understand that there is lots going on in there, and what we actually call it is a fluid mosaic model. So fluid mosaic model made up of lipids and made up of proteins that have several functions. So, and we'll go over again what is phospholipids, but remember we did talk about that when we went over our macromolecules. And so why is it called a fluid mosaic model? Fluid because the membrane is not rigid and it has the ability to kind of move around, so it's like more fluid-like. And then mosaic because it has a diverse range of different proteins that are found within the membrane. So we kind of call this mosaic because there's a lot going on in there, a lot of different and diversity. So the phospholipids. So our membranes are made primarily up of our phospholipids, and then there are proteins and other things that are embedded into the membrane. So reminder that the phospholipids are a group of unsaturated fatty acids. So because they're unsaturated, They have double bonds that are found in the tails of the fatty acids, and so that actually creates kinks in the phospholipid and prevents the phospholipids from packing too closely together. And if they did pack too closely together, it would be a solid at room temperature, but because it has the kinks in it, it remains a fluid. So it keeps it in the liquid form, which is what we want it to do, because we want things to be able to kind of move through the cell membrane. And if it was a solid, then it wouldn't really be able to have that ability for molecules to move in and out of the cell. So this is a really important word. It's called selective permeability, and that is a major characteristic of our membranes. So selective permeability is referring to this idea that some substances are able to move across the membrane more easily than others. And it's very specific about which ones can actually move in and out of the cell membrane. So for it to just pass through the membrane, there are a few things, and we'll be talking about what can actually pass through the membrane. It's usually really small molecules that are uncharged can move through the membrane quite easily, but there are other molecules like glucose that we need to get into the cell that are just really big molecules and they can't fit through. So they need the help of proteins. So these blue spots are proteins that will actually allow certain things to move through the cell membrane that are maybe too big to fit through the phospholipid bilayer. So we have these proteins that will help out. So here's another image looking at the complexity of our cell membrane. There's a lot going on in here, right? We have our phospholipids that make up the cell membrane, and then we have a whole bunch of proteins, different types of proteins that are found in here, and that all have these different functions and different abilities to do different things. So we have some that are more like a channel, and so a specific, and once again, it's very specific, molecules are able to bind to this channel and then they can fit through here. So not all molecules can fit through there, only specific ones can fit through. This is actually one that's transporting something out of the cell, and we can see here as ATP. We haven't really talked about ATP, but ATP is an energy source, it's a form of energy. So in order for us to, or for our cells to transport, let's say this red molecule out of the cell, it requires energy. This one didn't require energy, just easily was able to go through. And we're going to talk about why does one require energy and one does not require energy. And we're going to look at those differences. So here are the proteins, are some functions of our proteins. There's lots of functions to our proteins, and they're really all quite important. So we have to be able to maintain our cell shape. So having those proteins embedded into the membrane prevents it from becoming a solid, and then once again allows it to maintain that flexible shape. There's something called signal transduction. So there's these chemical messages that need to be sent from the inside of the cell to the outside of the cell, and so there are specific proteins that allow these signals to be sent from the outside to the inside and vice versa. There's enzymes. Enzymes are things that are going to catalyze reactions, things that will speed up reactions that your body needs to perform. There's also something called cell-to-cell recognition. So if we look back to, actually this slide is a little bit better for that, we have these little pieces right here. These are called glycoproteins. And glycoproteins actually have this carbohydrates that are kind of, you can see that's coming off of a protein. And that is a way for a cell to know that, yep, these are my cells, because it has, we call it a tag. It has a identification tag on the outside of the cell so that the body knows, don't attack this cell. These are your good cells. But if, let's say, a bacteria, a foreign invader comes into your body, doesn't have the same glycoprotein, it has a different glycoprotein, then the cell knows that this is not my cell and I need to attack the cell to get it out of here or destroy it. So that's what's very useful for cell-to-cell recognition, and also for transport. We had to transport things in and out of the cells or across the body. So that's another major function that the proteins will perform in our cell membrane, and we're going to be talking about more of those things as we kind of go through this unit. Okay, all right, so that is it for our cell membranes. I hope it was helpful, and if you have any questions, just let me know. Bye.